Hi everyone, welcome to Basic Science Series. I'm your host Lokendra Kumar, and today we will discuss toll-like receptor signaling pathway. As you can see on your computer screen or the device that you're using right now, maybe a phone or something else or tablet, what you're seeing right now is I have designed this particular signaling pathway that I will be discussing in this video. In this video, I will discuss how toll-like receptors they get activated and how the downstream signaling cascade transmits the signal to the nucleus where the transcription of pro-inflammatory cytokines and interferon production takes place. So it's a very interesting pathway where there are a number of molecules. You can see there are so many molecules. They are also known as adaptor proteins. They are involved in the signaling, and you can see there are certain distinct pathways that I'm trying to show you. Basically, in this video, what I'm going to do is I will discuss toll-like receptor signaling, activation, and then how the downstream signaling takes place. And also, if you haven't seen my previous video that I made on toll-like receptor structure and function, I highly recommend you to watch that particular video because that is the basic knowledge regarding the toll-like receptors structures. And while we are discussing the activation of the toll-like receptor, we need that information to understand clearly. Okay, so I want you to watch that video. After that, you can watch this particular video and you will be able to understand what I'm trying to explain here. Okay, all right, so with that note, let's jump on to the presentation part. A small recap of the previous video where I have tried to explain the structure. As you can see in this presentation slide, what I'm trying to explain is how human cell or any animal cell communicate with microbes. And there are these particular receptors, and in this case specifically toll-like receptors, they are involved in signal transduction, and there are different classes of these toll-like receptors. The important point that I'm trying to make here is you need to understand that what are the different classes of toll-like receptor and what are the ligands that are associated with these specific classes. So in that particular video, I have explained everything, and you can watch that video and try to understand the structure. All right, let's move on to the second slide. Here in this slide, you can clearly see I'm trying to show you the zoom in structure of toll-like receptor, and both of the toll-like receptors, in case of plasma membrane toll-like receptor and endosomal toll-like receptor, they have same mechanism, that is the mechanism of dimerization, where they form this M-shaped structure. Okay, so I'm just trying to show you when there is a dimerization, and dimerization is initiated by ligand, and there are specific ligands for specific toll-like receptors. Okay. Let's see what I have in the next slide. In next slide, what I'm trying to show you is the schematic representation of specific toll-like receptor 4. Let's say we have this toll-like receptor 4 present on the plasma membrane, and we all know, right? I'm sure that you have remembered the ligand for different toll-like receptors, and for toll-like receptor 4, the ligand is lipopolysaccharide. Okay, so here is our toll-like receptor 4 present on the plasma membrane, and what happens next is when lipopolysaccharides combines with MD2 protein, MYD2 protein, the full form of MYD2 protein is myeloid differentiation factor 2. And this particular protein, in combination with the lipopolysaccharide, there is another protein that is also known as LPS binding protein. It is present in the serum. So this particular complex initiates the dimerization of the toll-like receptor 4, and there is another additional protein that contributes to the activation. This is CD14. CD14 is the cluster of differentiation 14 protein that is involved in this activation. And here you will get a complex formation of TLR4 LPS MD-2 MYD-2 and CD14. So this is the full complex that initiates the dimerization as well as the signal transduction through TLR4 receptor. Okay, so now at least you know how the activation of the toll-like receptor takes place on the plasma membrane. Let's move on to the next slide. As you can see in this slide, I'm trying to show you that this particular domain, this domain is also known as TIR domain. If you have correctly remember what I have explained in my previous video where I've tried to focus on this particular domain, and this is the zoom in structure of this domain, and as you can see, this is the dimeric form of TIR domain of specific toll-like receptor. And you can see that because of this interaction is really important for the signal transduction. And what is TIR domain? This TIR domain is basically interleukin-1 receptor family member, and it's also known as TIR domain, which I've already explained. And the structure of this particular TIR domain is unique. It has central 5 parallel beta sheets. You can see these sheets, right? These arrows, they are beta sheets. And then you have specifically 5 alpha helices. So you can see these helix structures, they are present. 5 beta sheets and 5 helical structures are present in this particular protein. And there are certain conserved residues that are present in the hydrophobic core of this particular molecule. So this was the just brief introduction about the TIR domain, and because of this interaction you get that signal transduction transmitted to the adapter proteins. Okay, so now let's start the signaling process. We already explained, as you can see here, I have designed that structure, and it's just the illustration. Okay, I'm just trying to show you all the components, and you can see when this whole complex, the activation complex, specifically I'm talking about TLR4, is constructed, there is the activation of these adapter proteins. So you can see on your computer screen that I have shown you different adapter proteins. Let me also show you the names of these proteins, so it's easier for you. Okay, now you can see the protein that is initially involved in case of activation of MYD88. So here I want you to understand that there are 2 pathways that are initiated after the activation of the toll-like receptor 4. One is MYD88 dependent pathway, and second one is TRIF-dependent pathway. Okay, and I will explain to you what are the molecules that are involved in these 2 different pathways. Here I'm trying to explain, you see, we have plasma membrane toll-like receptor as well as endosomal toll-like receptor. They have a similar kind of initiation mechanism. They also activates through MYD88 as well as the TRIF dependent pathways. So what is MYD88, right? So you can see here that MYD88 is myeloid differentiation primary response gene 88 is the name of this particular protein. It's a universal adapter protein for most of the toll-like receptor except TLR3. So this is an important information for you, specifically related to the TLR signaling pathway, that TLR3 is independent of MYD88 dependent pathways. Okay, now another protein that helps in the activation of this is TRAP, which is toll interleukin-1 receptor domain-containing adapter protein. Because it has this interleukin-1 receptor domain, so it's known as TRAP, and it also helps in the activation pathways, right? So and the next protein you have which helps in the activation of TRIF, that is TRAM. Okay, so you have TRAM protein, which is TRIF-related adapter molecule, because they are related to the TRIF pathways. That is why they are known as TRIF-related adapter molecules. They have long names, but try to remember these proteins because they are important when there is question related to the TLR signaling pathways. What are the proteins? What are the proteins involved in MYD88 or TRIF pathways, right? And while you're doing research, you also need to know, if you are trying to understand the signaling, you need to know the expression of these proteins. If you are studying a specific pathway related to a specific ligand molecules. Anyways, so as you can see, I have designed that there is a initiation of MYD-dependent as well as initiation of TRIF-dependent pathway. Let's move on to the next slide. Okay, so this is my next slide, and I'm trying to show you when this MYD88 molecule or MYD88 adapter protein is activated. You can see IRAK4 and basically IRAK1, IRAK2, IRAK4 protein, they are all proteins, they are activated, and they lead to the activation of TRAF6 protein. Okay, and in this case there is also an indirect activation of TRAF6 through this specific protein, TRIF protein. Okay, and what is TRAF6? TRAF6 is basically tumor necrosis factor receptor associated factor 6. So it's a long name for this particular protein. And you know what is tumor necrosis factor? It's one of the pro-inflammatory cytokine. And what is the full form of IRAK4? IRAK4 is interleukin-1 receptor associated kinases. And now we all know what are kinases. Kinases are the enzymes that transfer phosphate group from high energy donor to any substrate. That leads to the activation of this intracellular signaling pathway. Okay, so and you will see in most of the signaling pathway, you will see kinases, they are involved and they are activating and they are phosphorylating the molecules that are involved in the signaling. Here you can see this similar kind of complex is working right here, and that is also helping in the activation of TRAF6 molecule. Okay, now you can see I've just explained to you the next step for the MYD88, and I have explained to you what are the molecules that are associated. Let's move on to the next slide. Okay, here you can see when TRAF6 is activated, there is an activation of these particular important proteins, that is MAP kinases and NF-kappa B. And NF-kappa B as well as MAP kinase activation leads to the pro-inflammatory cytokines production. This is an important point to remember, that whenever you have MYD88 pathways that are activated, they mostly helps in the production of pro-inflammatory cytokines. In case of TRIF protein, you will get the interferon production. Okay, so this is important. And interferon, they are involved in the antiviral mechanisms. I will explain to you in my next slide. But I have certain points that I want to show you here. There are some inhibitor molecules that stop the activation of this nuclear factor kappa B protein, and the full name of this protein is basically nuclear factor kappa light chain enhancer of activated B cell. So that is the full form for NF-kappa B protein. And when it is in the resting stage, there is some kind of inhibitors that are bound to this particular protein. And the mechanism of nuclear factor kappa B activation is a very detailed. And if I try to explain the full mechanism, I mean, this is going to confuse you. So I'm just trying to tell you that there is some inhibitory molecules that are attached. And because of this activation pathway, you will get nuclear factor kappa B activation by degrading those inhibitory molecules. And then you will get the transcription of these particular cytokines. Okay, so I just wanted to introduce you to the signaling and not going into the very much detail of the NF-kappa B signaling, but I will definitely make videos on this specific signaling system because it's very, very important in case of any intracellular signaling pathways. All right, next is, as you can see here, I'm trying to show you that there are these kinases that are involved, and I have already discussed the function of these kinases. And then TAC1 is another adapter protein that helps in the activation of NF-kappa B as well as MAP kinase. And what is TAC1 is transforming growth factor B activated kinases, and this is also the kinase molecule, kinase enzyme, and that is why we call it as a kinases that helps in the activation of this particular pathway. All right, and you can see the similar mechanism of this activation takes place in this case, right? Let's move on to the next slide where it is clear. So you can see here what I'm trying to show you. This is your MYD88 dependent pathway, and this also works in case of your endosomal toll-like receptors. So you can see you get TRAF6 activation, you get TRAF6 activation here, you get nuclear factor kappa B activation, and MAP kinase activation here, right? And then that leads to the pro-inflammatory cytokine production. All right. Now, in case of TRIF pathway, you will see that there is another adapter protein, that is TRAF3. So TRAF3 protein helps in the activation of interferon production. So through IRF3 and IRF7 activation, okay, and there are these kinases molecules that are involved in the phosphorylation, and these proteins are important. And what are these? These proteins are similar to the adapter proteins of MYD88 dependent pathways, right? And they involved in the interferon gamma production. So basically you have two pathways, one MYD88 dependent pathway and second one is TRIF dependent pathway, and you can see both are leading to different effector molecules. In case of MYD88, you will have pro-inflammatory cytokines, and in this case you will have interferon production. So it seems like for bacterial antigens you will get this particular pathway activated, and in case of viral attack, viruses, you will get this particular pathway activated. So you can, on the basis of your requirements, specifically in research, you can study these specific pathways. And I have mentioned the full form of TRIF. You can see TIR-domain-containing adapter inducing interferon B, because they induce the interferon production. That is why they are known as adapter inducing interferon beta. So full form of TRIF. Okay, so this is basically the general layout of toll-like receptor downstream signaling. Let's summarize what we have learned in this presentation. Is, in summary, I can tell you that first there will be the activation of toll-like receptor 4 by lipopolysaccharide, which is, I have mentioned here, LPS. Then you will have MYD2 protein as well as CD14 cluster of differentiation 14 molecule, and that particular complex formation will lead to the specifically activation or dimerization of this particular molecule, TLR4 in this case, and then TIR domain, which is present inside the cell, will form the dimer, right? And that activates these two specific pathways, MYD88 dependent and TRIF dependent pathways, which leads to the activation of MAP kinases, and nuclear factor kappa B activation also leads to the production of pro-inflammatory cytokines as well as interferon production. So as you can see, this is basically a crux or a summary of the pathways, and I have already laid out this particular pathway. And if you try to read this particular signaling pathway, you can, Google Scholar some of the review articles, and you will get multiple articles on this specific toll-like receptor signaling, and most of them will have these kind of signaling system explained in detail. Okay, I have just explained the basic layout of the toll-like receptor signaling. In this case, I took example of I have taken the example of TLR4 molecule, right? And this is how these are the proteins that are involved, and most of the proteins, they are complex proteins, and there is a complex regulatory mechanism that involves in the activation of that particular protein. And specifically my favorite is NF-kappa B activation, and there are canonical, non-canonical pathways of NF-kappa B activation, really interesting pathways. So we will discuss those pathways in future, but right now I want you to remember that toll-like receptor signaling involves these proteins and what are the names of these proteins, right? And is there any similarities and is there any difference between these two pathways? All right, so with that note, I will just conclude this presentation, and I hope now you have a good idea about the toll-like receptor signaling pathway, and you can see it's a very simple pathway where you have activation and then transmission of the signal. In the next video, I will bring you more interesting topic related to the immune system, related to microbiology, related to general biosciences. That's what I do in this channel, and those videos are definitely going to help you. In case of this particular video, you can make notes on these specific signaling pathways, and you can read more about these specific signaling pathways because in this video I have not covered in very much details of the certain aspect of the signaling pathways, right? So this is just a starter. I have given you the starter. Now this is your responsibility to study in detail and make some notes on these specific signaling pathways. All right, so I will meet you in my next video where I will discuss same kind of interesting topics related to biosciences. If you like the video, please press the like button and also subscribe to the Basic Science Series YouTube channel. Thank you and take care.